This is Lab 5, Insulin Signaling. To complete this lab, you're going to need to download the PDF and read it. And the PDF is attached under the assignment where it says Lab 5. I have also made a Word document, and that's the document I actually want you to submit. And I've just kind of copy pasted a whole bunch of questions onto that Word document from the PDF. And those are the only questions you have to answer, the questions on the Word document. Okay, so here we go. Insulin signaling lab 5. Firstly, let's consider the fact that we are multicellular organisms, and so we're composed of all these different cells. So here I've drawn 1, 2, 3, 4, 5, 6, 7 cells. All these 7 cells, and for them to work together to form an individual called you or me, they need to communicate with each other. And they do that by secreting signaling molecules, which you would have learned in the lecture this week. So a couple of spaces that we need to be able to name. Number one, we can already name the cytoplasm. That's in cells, so that would be right in there, right? That's where the cytoplasm is. And then the other space that we need to be aware of, we could call it the, your book calls it the interstitial fluid, sometimes also called just the intercellular space or intercellular fluid, sometimes extracellular fluid. Good grief, so many words. We're going to go with interstitial fluid. So any molecule that is going to be going from one cell to the other is going to have to go through that. But there are also other things other places that are between cells and can help cells communicate with each other, such as, hey, holy smokes, somebody drew some capillaries. They're really good at drawing, aren't they? So here are some capillaries in red, and this is where blood would be flowing like that, right? Blood's flowing like that. And the blood transports a huge variety of things. What we're going to be talking about today is blood glucose. So blood glucose is transported in the blood, and it's really important to do a couple of things with blood glucose. Number one, it needs to be maintained at a relatively, not a constant level, but within an upper limit and in a lower limit. So it'll go up, it'll go down, it'll go up, stay high, then maybe crash, and you feel tired and grumpy, then it comes back up. But it must stay within certain limits because it has effects on cells. Next, all cells require glucose. Well, I don't like to use the word all cells, but cells really like to use glucose for cellular respiration to produce ATP. It's like the gasoline of the cell world. So let's draw some glucose in here. Whoa, looks like this thing got like chicken pox or something. No, but those are glucose molecules. So we're just going to think of these little fellas here as glucose molecules, glucose molecules. So what happens when you flood the system with glucose, say you just ate a sugar cookie or a big bowl of rice or a potato, all of that gets broken down into glucose eventually, and it goes into your blood, and that can have some negative effects. The lab lists a whole huge number of effects, of consequences for not being able to regulate your blood sugar. I'm just going to go over one here, though. So let's consider there we go. The solute concentration of the blood here, solute concentration of the interstitial fluid, and then, of course, the solute concentration in cells. So what happens when you have a whole bunch of sugar in the blood, we're going to just be thinking about this one for now, is, well, the solute concentration is going to go up. Ooh, it's going to go up. So the solute concentration goes up. So what does that mean? Well, let's remember what osmosis is. Osmosis is the diffusion of water across a semi-permeable membrane, diffusion of water down its concentration gradient. So if we have an increased solute concentration in the blood, what that means is we are going to have a loss of water from the interstitial fluid. See, you don't believe me? Well, look, the computer even agrees with me. That red arrow, right? So water diffuses out of the interstitial fluid. Now, what does that do when the interstitial fluid right here is losing water, that causes the solute concentration to go up, as we can see with this arrow. Thank you for agreeing with me, computer. So solute concentration goes up in the interstitial fluid here. What that means is has more consequences for water movement from cells. Now the cells start losing water as well. And this is when the cells are getting dehydrated. The cells are losing water, and this is a huge problem because water is what everything else exists in. Everything else is dissolved in the water. And so if there's less of it, it affects basically every metabolic reaction you can imagine, and eventually somebody will die. People with diabetes sometimes can wind up with frequent urination. And so why is that? Well, we can see water is being lost from the interstitial fluid here. It's going into the blood. That can raise your blood pressure. That's another bad thing. And then the body responds to this higher blood pressure by going to the bathroom. So this extra water goes to the kidneys where it is lost in the urine, with the urine. So really important to regulate how much sugar there is in the blood, how much of this. So we have to do something with it. When blood sugar goes up, it's got to go somewhere. So one of the things, so a really good thing to do with blood sugar is to get it into cells, because cells are happy when they have sugar because it means that they can do aerobic respiration and make ATP. The liver can also absorb blood glucose. It'll convert it to glycogen, which we remember that's a polymer of glucose. It's a nice storage method. Muscle cells will also help to regulate blood glucose. It's maybe one of the reasons why some people might say that bodybuilding can be healthy, very healthy even. That muscles help to regulate blood sugar. They can, getting a little bit out of my realm of expertise there, but anyway, cells absorb blood glucose. So let me go to the next slide and we'll take a look at how blood glucose changes throughout the day. Oh, there's one more arrow I forgot there. Okay, so control of blood glucose throughout the day. Let's take a look down here. You wake up, your blood glucose is low, and that might be why you're feeling a little bit low energy, so you decide to have breakfast. Yay, you've eaten. Most breakfasts are going to involve some type of carbohydrate, and if yours did, then you see your blood glucose is going up. Now, what if it just kept going up and up and up and up and up? Well, we saw what the consequences are, right? You get dehydrated cells, water is leaving the cells. So when blood glucose gets this high, a part of your brain, you don't need to memorize it, but the, what is it? The pancreas, never mind, it's not part of your brain. The pancreas measures blood glucose, and when it's high, we get insulin secretion. Insulin secretion is going to happen right around there. Why do I know that? Well, because now the blood sugar is going down. Insulin causes cells to uptake glucose, right? So if this is a cell and insulin has, I'll draw a little receptor here. Okay, insulin's bound to its receptor. Somehow all these molecules of glucose are going to start diffusing into the cell, and then the cell gets a little smiley face because it's getting glucose. So when insulin is secreted by the pancreas, we get a decrease in blood sugar. That's great. So this is cell-cell signaling. The pancreas secretes a signal, insulin. It's a hormone, and it is transported in the blood. From the blood, it'll diffuse out of the blood everywhere into the interstitial fluid, and then it will bind to receptors on its target cells, causing them to uptake glucose, causing blood glucose to go down. All right, so what happens is eventually blood glucose is really low, and this second hormone, the lab doesn't even talk about it, but you guys are certainly going to ask, well, what about when it gets low? Well, there's another hormone called glucagon. It's really easy to remember because it's like gone, right? The glucose is gone, and so when that one's secreted, it causes certain cells to release glucose into the blood, so blood glucose will go back up. Specifically, the liver will release blood glucose, and that'll get you all the way until lunch. So the liver has released a bunch of glucose here; it's going up, and then the cells are using it, of course, so it's going to be going down. Insulin might be playing a bit of a role in helping those cells to absorb glucose. Anyway. Eventually you get hungry because your liver is running out of glycogen, and for a variety of other reasons. And so what you do is you go eat, and then your blood glucose goes way up again, and then insulin secretion happens all over again, and blood glucose goes back down. All right, so it's going up, down, up, down. And you can just forget that word glucagon if you really want to. It just causes blood glucose to go up. For this lab, we are going to be interested in what does insulin do when it binds to a receptor on a cell, in the cell membrane. We talked about already a little bit in lecture as well, but we're going to go get a lot more details now. Okay, this is a video, and it's got a lot of details in it. Don't worry about memorizing all the details in this video. The details you need to know are in the actual lab, but I thought this is a pretty good visual aid, so I'm going to play it. If it's going too fast for you, you might want to review what we know about all these receptors. There's a diagram in your book that involves PIP2, DAG, IP3, release of calcium, stuff like that. And this is basically what that is. So I'm going to play it now, and you can skip it for now if it's a little bit too much detail, and then come back to it later if you want.
A biological individual consists of multiple organs with specialized functions. For the organism to function properly in its environment, these organs must communicate. This communication often involves a signal sent from one location to another that instructs the second organ about the status of some cellular feature. Glucose is a good example. Glucose is a critical product of digestion. It is an essential energy source for cellular metabolism. This energy is produced when glucose is used as a substrate for glycolysis and then the Krebs or citric acid cycle. Following the digestion of food, higher levels of glucose circulate through the bloodstream, where it enters different cell types. In muscle cells, glucose is readily used to produce energy and is also stored as glycogen, a secondary short-term energy source. In fat cells, glucose is used for triglyceride production and acts as an important energy reserve molecule. Here, we will illustrate the signaling pathway that occurs when glucose is at high levels. This pathway involves multiple proteins and signaling events. This is termed cytoplasmic signaling. Different types of cells perform similar signaling steps in response to changes in their environment. In the protein recycling animation, we see a group of storage vesicles enriched with GLUT4 proteins continuously recycling from the cell membrane to an inactive location in the cytosol. GLUT4 is a protein that facilitates the movement of glucose into the cell. When high levels of glucose are detected by beta cells in the pancreas, insulin is released by the cells. The insulin circulates through the bloodstream until it binds to an insulin receptor embedded in the cell membrane of a muscle, fat, or brain cell. Once the insulin binds to the receptor, phosphate groups are added to the intracellular domain of the receptor. Since the receptor itself adds the phosphate groups, the process is called autophosphorylation. This phosphorylation event sets off a cascade of molecular events. The activated receptor protein then adds a phosphate group to another closely associated protein. This effectively passes the signal from the receptor to the next step in the signal pathway. Proteins that add phosphate groups to another protein are called kinases. Kinases are often components of signal pathways, and phosphorylation is an important component in the transmission of a signal from one compartment to another. In this system, the signal corresponds to the level of blood glucose and is transmitted from outside to inside the cell. Next, we see a large pool of molecules that are embedded in the membrane, also being phosphorylated. Other proteins are then in turn phosphorylated, further transmitting the first extracellular signal that was originally sent from outside the cell membrane. So how does this affect the uptake of glucose? As we mentioned before, GLUT4 is a glucose transporter, and GLUT4 storage vesicles are held in a recycling state near the cell membrane. The vesicles are held mostly in this region because the Rab proteins that interact with the motor proteins necessary to move the vesicles to the membrane are in an inactive state. The final step in this signal pathway involves the phosphorylation of a protein that prevents the Rab proteins from interacting with the vesicles. When the Rab proteins are no longer inhibited, the storage vesicles can freely merge with the membrane. Once the vesicles have merged, many GLUT4 proteins are embedded in the membrane, and large quantities of glucose can move into the cell. It is the signaling pathway that ensures only the correct molecules will be allowed to enter the target cell.
Okay, so this last point here is really critical in that it sort of brings together a lot of stuff that we've learned about. So what's going on here is these purple fellas on the right there, those GLUT4 transport proteins, that would be facilitated diffusion. They're allowing a large molecule glucose to diffuse into the cell, and they were stored on vesicles. So remember, those vesicles would have come from either the Golgi apparatus or the rough ER, and any proteins embedded in the membrane of a vesicle will become embedded in the membrane of the cell if the vesicle fuses with the cell membrane. You might want to replay the last little bit of that video if you didn't quite catch that. I think we've got maybe another 10 seconds on the video. Oh, well, that was exciting. Okay, so you can go back to that if it was a little bit fast for you. Okay, so now we are to the last slide in this presentation, and let me just circle the title. It's Figure 4. Pretty exciting, huh? So Figure 4, this is from the PDF that you're going to have to download from our Blackboard course website. It's on page, I can't remember, something like page 3 or 4 or something, and it says that it wants you to label all these parts of this diagram. I'm not going to ask you to do that because I just want to save you a little bit of time in terms of like taking a picture of your labeling, then copy pasting onto the Word document. But let me say another thing. You do need to understand what this diagram is. So I'm not going to ask you to label it all like they've asked you to, but you need to understand what this is. So let me just go over it a little bit. Number 1, so the signaling molecule is, of course, insulin, and it is a tyrosine kinase linked receptor. There's 2 of them, 1, 2. And when we get 1, 2 insulin molecules binding, each one binds to its different receptor, they undergo a conformational shape change and they come together, forming a dimer. Then the inside, well, the whole receptor undergoes a conformational shape change, and that causes phosphorylation. See, right here. There's a phosphate, and then there's another one here, and then there. And there would actually be another 4 over here. 1, 2, 3, 4. They've simplified it a little. That, in turn, leads to the activation of a whole bunch of proteins. Activation of them, what does that mean? Well, they're going to be phosphorylated pretty much always. When a protein is activated or deactivated, it means that a phosphate group's been added or taken off. Over here, all of these, MEK, MAPK, ERK, and then ELK, these are all just examples of different kinases that will lead to causing different cellular responses. Different cell responses. So these are all kinases, and they lead to different cellular responses. They can be involved in signal amplification as well. Here they've called it just signal transduction. But having this cascade from one to the next, that can also amplify the strength of the signal. Okay, so some of the responses are these TFs. That stands for transcription factor. And what transcription factors do is they go to DNA and they will bind to the DNA, and that will either turn on a gene or turn off a gene. In this case, we can see that mRNA has been produced, And again, M stands for messenger, messenger. So somehow this transcription factor here went into the nucleus and caused this messenger molecule to be produced, and that's going to diffuse out of the nucleus, will be transported out of the nucleus, and then do whatever it does. It's some kind of a gene. The DNA has got the gene. It is made into mRNA, and then it goes out of the nucleus and is probably going to be made into a protein. And who knows what response that will be. We're not interested in those specific details. The vesicle that has these GLUT4 proteins on it is right here. There it is. It's a vesicle. And this is a phospholipid bilayer. This is a phospholipid bilayer. And that means that it can fuse with the cell's membrane. And when that happens, these GLUT4 carrier proteins will become embedded right here in the cell membrane. And that increases the permeability of the cell to our good buddy glucose here, which can now diffuse into the cell. Result is less glucose out here, right? Less glucose out here, and then in turn less glucose in the blood, and the cell inside is happy. Why? Because it's got glucose, and glucose is a great source of energy. Okay, so you got to understand this slide, and they describe it further in the PDF, but you don't have to label it and submit that. It's just going to be a whole bunch of questions about the reading on that PDF. You have to take a look at the Word document. It's fairly straightforward. Look at the Word document and look at the PDF, and you got to read them both. Basically, let me just say one more thing, the kind of housekeeping. How do you do this assignment so we can minimize confusion? Basically, there are a few different parts. I think, what was it, 3 different parts. So what you want to do is, so first read part 1. That's in the PDF. Second thing is I would answer the questions, answer Q's for part 1. And this is all labeled in the Word document. It should be pretty simple. You just read part 1, then answer the questions that are labeled part 1. And then 3, you're going to read part 2, and then answer the questions for part 2. I'm not going to continue writing this. It's pretty simple. Also, for this assignment, I encourage you all to post as much as you want about what you think the answers are to these questions. You can, it is actually meant to be a bit more of a discussion. So I'm going to bring the discussion board back online as soon as our test is over, and you guys can absolutely post one of the questions, copy paste it in there and say, this is question 2 for part 1, and I'm completely confused. Can anybody help me? You can even say what you think the answer is. So grading on it will be a little bit more like done, not done. Did you attempt to read the text well and understand it, and then write a complete response? The quiz is more where you'll be getting assessed for how much of it you actually understand, and that's just because allowing you guys to talk to each other very, very completely about the answers. So what you think the answers are for these, you can feel free to share those or ask for help on the discussion board. So visit the discussion board, ask for help. I may even answer your questions myself. I kind of like you guys to figure it out yourselves, but I'm of course here to help. And so if you really can't answer one, just ask me and I'll even answer 1 or 2 for you. All right, so I hope you enjoy this lab. It is about diabetes. It's a kind of learning activity called a case study, where we look at sort of a real life example of how we do science. So I hope you enjoy it, and best luck to you. Let me know if you have any questions.